So as I saw the other talks today, as I saw Jan's talk, I was like, oh, I should have talked about the Ms. Pac-Man work coming out of Montreal. But instead, I'm going to talk about some of the things that are more, they absolutely require ML at scale, but I'm going to also talk to you a little bit more about the science that goes into them. Because for me, I think that biomedicine is just going to be the most important application of ML at scale, because it's going to change healthcare for all of us. Okay, so these are a few problems that people in my lab and other parts of Microsoft are working on. The first is CRISPR gene editing. I'm sure you guys have heard about CRISPR. It's a way of editing genes so precisely that we hope we'll be able to go into living cells of people who have a disease and actually get the bad DNA out of there. So it's really interesting, and it has required ML at scale. That one is pretty much wrapped up. I mean, I'm sure there'll be more things, but that one's pretty much wrapped up. This one I will only talk about in passing. I'm super excited about it, though. It's a partnership of Microsoft Research and Stand Up to Cancer. Stand Up to Cancer brings together about 1,500 researchers and 150 institutions. Our project is called the Convergence 2.0 project. It's bringing machine learning to cancer immunotherapy, and there are about another half dozen places involved in this. In particular, in Stanford, we're working with Mark Davis and his group. Okay, but I'm not going to talk too much about that, except that what I will also focus on for about half the talk is decoding the immune system. This is a partnership with a biotech. It's called Adaptive Biotechnologies, and this is taking your immune profile and trying to diagnose what diseases you may have. And that's super relevant for cancer, because we may be able to tell you, oh, you have pancreatic cancer brewing, long before you would actually find the pancreatic cancer, which is usually too late. Okay, so for the CRISPR, here's the team. It was Jen and Nicolo out of the Microsoft Research New England lab, some people at the Broad, and several other places. Okay, so you have probably seen that gene editing is getting a lot of attention over the last few years, and also a lot of hype over the last few years. Sure, all of you have heard about it, but I know that a lot of people don't know how it works. Turns out that the Brain Institute at MIT put together a really wonderful, minute and a half little clip on how it works. And they've told us that we can use it, so here it is. Are we hearing it? We are not.
Changing genes in living cells is not easy. But recently, a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target.
Okay, so what we want to do is figure out how to match the target, how to come up with the guide DNA. The guide DNA is 23 nucleotides long, not very long. So how do I choose it? It turns out that if I want to knock out one gene or one transcript of one gene, there are 200 places over the gene where I can do it. How do I know the best one, the most effective one? So that is the on-target problem. And our team was the first team to bring ML to that problem. And then the off-target problem is so much worse. Once I tell you this is the right guide to cut where you want precisely in that gene, then that same 23 nucleotides could screw up something in the other 3 billion nucleotides in the human genome. So how do you deal with the off-target problem and score how bad it might be, how bad the collateral damage might be? Okay, so the solution paths, so what people first thought about was let's do more wet lab stuff. The Broad had been doing a lot of wet lab stuff, but they had 6 guides and it took them 3 months of wet lab work for 6 guides, okay? They had collected a lot of data, using these kinds of little wet lab experiments. So then we came in with ML. So what do you do here? Well, so you saw this picture of this little Cas9. So what I want to do is I want to look at my RNA guide. This is 23 nucleotides. It has certain features, like what nucleotide is in what location, like how likely that guide to help in the unzipping of the DNA, and this has to do with the melting temperature, how close it is to the end of the gene, because things don't work as well when it's at the end of the gene, that kind of thing. Came up with a model that predicted the success of the gene edit. And it was a regression tree, a boosted regression tree model. Okay, the problem scale, there are 40 million possible guides, and this is because we have 20,000 genes. The Broad wanted to come up with a library that anyone could use. There are roughly 10 transcripts per gene. That's the same gene can be transcribed in many different ways to get different proteins, so that's another factor of 10. And then there are 200 positions per transcript. Okay, so the result of this boosted regression tree analysis was a 20% improvement in accuracy over what had been done just with wet lab work, and a 50% savings in cost. There's been strong uptake, so this came out a couple of years ago, and it's basically what everyone in biology uses now. You can just call it, it's called all the time. Okay, the much harder problem, and the place for which you really need scale, is the off-target problem. Okay, as I said, and I should mention that our collaborators at the Broad said, it is absolutely impossible. No one could ever do the off-target problem. But of course, if you want to insert a guide into living cells, okay, you really want to know what kind of collateral damage it might cause. You may choose a much less good guide at cutting where you want to make sure that it doesn't mess up really important parts of the genome. So there are 3 billion nucleotides across the genome. There is only sparse training data, and this is true of a lot of biomedical problems. Not images, okay? Images, you do really well with DNNs, okay? But in many biomedical problems, there's only very, very sparse training data, either because you're doing drug trials, you have very few patients, very high dimensional, but very sparse. In this case, you can only measure genes which have a direct observable effect, but it could be that you're messing with something that is not even a gene, but it has a really important regulatory effect. There's a combinatorial explosion, so every one of these off-targets is a mismatch. At least one nucleotide is off, okay? And so it's matching somewhere else in the genome. But if we go up to, let's say, 6, which is what we do, mismatches, there's a combinatorial explosion. And also, if I get 1,000, 2,000, 3,000 of the most devastating off-targets, Do I just return to a biology researcher who says, what should I cut? I'll say, oh, good, this is the on-target you should use, and here are scores for each of the 2,000 off-targets. Figure it out. No, I want to give them one off-target score. Okay, so here's a combinatorial explosion. This was for a biology audience. I think you guys know how to do, so 69, where does that come from? 23 nucleotides in the guide, and each one of them, if it were an A, it could have been a T, C, or G for a mismatch, so 3, so 23 times 3, and this is for a biology audience so that you can see what happens with 23, choose K times 3 to the K, okay. So the one full example is totally done in the wet lab, but then there's very sparse sampling of the other ones. So you really need to do a lot there. So there's a 3-step approach that our researchers took to the off-target modeling. The first, and here there was a lot of biology that went into it, was to filter the genome-wide potential off-targets to assess which, and we included only the 2,000 worst. If they're near the middle of the guide, they're worse, and certain kinds of ones are worse. So we came up for each mismatch with the 2,000 worst things that it could do. And then we scored, so this is where the ML came in. We scored each off-target for activity using a machine learning predictive model. And this was a part they thought was impossible. We pre-computed all of these, okay? And then we aggregated the scores from 2. So, so here are 2,000 off targets for the guide you think you want to use. Let me give you one overall score for that. But we have Spearman weight so that we can weight those 2,000 differently. I'm going to talk to you really just about the second one. Okay, so now, instead of just the RNA guide, which had these features which in the past were called X1 through XM, now I'm calling them G because they're the features of the guide. And here are the features of the off-target, okay? And we again learn a model. This is a boosted regression tree, which tells us what the activity should be. And high activity is bad here. I don't want it binding to some other site besides the gene I'm trying to cut. Okay, so what I do is I first start off with a single mismatch model. So here's one mismatch and I get an F1. Here's another mismatch and I get an F2. And then I want to learn something that puts them together. These are typically not independent, so I want to learn the dependent structure. Okay, it's not naive Bayes at all. So that is really the second level of the learning in the model, and that one is incredibly important. Now I have this for 2,000 off targets, and you might say, well, I could just weight them all equally. That's what you might do if you were going to do a wet lab verification anyway. If you were not going to do a wet lab verification, you might say, oh, these 5 look really the worst, so I have these Spearman coefficients which weight them up. And here is what happens. Here I'm changing those coefficients. This is if I'm weighting them all equally. This is if I'm weighting some much more than others. And these are the competing models, and they really did a lot worse. So there were these 80 billion pre-computations that we did. It took about 7 million CPU hours. But doing those now, what happens is that anybody, so this paper just got published a month ago, and if you're going to Nature, you can't put out any tools or anything until your paper is published. So now, CRISPR.ml, anyone can go there. You put in what gene you want to edit, and it will give you an on-target score and an off-target score for that, okay? And you might look over several of them because you personally say the on-target is more important or the off-target is more important. Okay, and this has only been out there for a month now. This has only been out there for a month now, but it's already starting to get a lot of usage from academic institutions, research hospitals, and it's just been out there for a month. But if you want to do gene editing, this is the way to go. Okay, Now I'm going to switch topics to cloud scale immunomics. If you haven't heard of immunomics, you will. I think this is going to be as important as genomics, actually, for healthcare. It's going to be incredibly important. And we are super lucky. We just entered into a partnership with Adaptive Biotechnologies, and they are the leading company for immunomics. And some of this also relates, in fact, the picture up there, which you can't see super well, is killer T cells eating a cancer cell, okay? Turns out that our immune systems are the reason that there are so few childhood cancers. We're all developing cancers all the time. And our killer T cells, our T cells are going after them and killing them, okay? As we get older, our adaptive immune system gets worse, and also the cancers get much more sophisticated. So I'll mention a little bit about this. This has to do with that Stand Up to Cancer project. This is much broader than that. We are going towards learning how to decode the immune system towards a universal diagnostic. Okay, so each of us at any given time has about 10 to the 12th T cells circulating in our blood, naive T cells, okay? Some of them, there are very few. Others of them, there are really a lot because, oh, I had the flu this year and I have T cells which know how to target that flu now. Or in some years, you just get the flu vaccine and it works. For many people this year, you got the flu vaccine and you still had to do it all by yourself. Okay, so what happens is that when you get a blood sample taken by your physician, she can take 1 ml of blood and she can send it off to Adaptive. And in that ml of blood will be 10 to the 6th T cells. Now in you've got 10 to the 12th T cells, but really the ones that, some of them have just never seen anything that they bind to, okay? So I uniformly sample by taking blood, but really what I'm going to get overwhelmingly are the T cells that have expanded to millions of T cells, okay? So those are the 10 to the 6 T cells that I tend to see. And what Adaptive will do is they will do the DNA sequences of the T cell receptors of those 10 to the 6th, those million T cells in your 1 ml of blood, okay? And the T cell receptors are the really working part of the T cell. That's the part that recognizes things, okay? And then what we want to do is put that together with machine learning in ways that I'll describe over the next few minutes, which will enable us to look at what T cells you have and say, wow, you've got Lyme disease. Wow, you actually seem to have this autoimmune disorder. And you seem to have the very beginnings of pancreatic cancer. And go in there and take care of these things before they get out of control. Okay, so here's the biology, which is, this is just so cool. This is amazing that we have evolved to do this. It really is. Okay, so your body makes these T cells. And identical twins, even when they're born and haven't been exposed to things yet, have completely different T cell receptors. Completely different. Because you were born with certain genes that you get from your parents, but in forming these T cell receptors, your body goes in there and just randomly splices these things and throws them back together. So every one of us is a factory for generating different T cell receptors. So, maybe I have a T cell receptor that my identical twin doesn't have. So there's a better chance evolutionarily that one of us will survive, okay? So it really makes a lot of sense that this would happen. Of course, since these things are generated randomly, a lot of them are going to react with self, okay, which is a disaster. That's how you get an autoimmune disorder. So in your thymus, all of them are exposed to self, and the self-reactive ones are thrown out, okay, hopefully. They're thrown out. And the others get released into the bloodstream. So here's a schematic of a T cell. The T cell receptor is the part that's different on all of them, okay? So what's going on in a cell? Well, in a cell, there are about 100 million proteins at any given time. Near end of life, these proteins get chopped up into little peptides, like 9 or 10 amino acids long, okay? And many of these get displayed on the outside. Many of these fragments get displayed on the outside of the cell, these complexes called MHC. By the way, you also have different, there are about 10,000 MHCs per cell. We also all have different MHCs. And this is very, very relevant. I mean, this will tell you maybe some, like cancer immunotherapy might, a particular cancer immunotherapy drug might work for Christian and not for me because he has different MHCs than I do. You get these from your parents also. You inherit like 6 or 7 HLA types, and it leads to these. Okay, so that MHC, people describe it kind of like a hot dog bun holding that little protein fragment, which is that little purple lilac thing, okay? And normally nothing happens because the T cell receptor has already gone through the thymus, been exposed to self, knows that little 9-mer, and says, oh, that's self. I'm not going to do anything. However, let's say that a virus comes in and it takes over that cell and it starts producing its own proteins, okay? And then those proteins get chopped up and those little 9-mers get put out and, whoa, maybe, 1 in 10 to the 12th, maybe that TCR recognizes that, binds, and it kills that cell, okay? And moreover, it expands, okay? Thousands and millions of them. So it reproduces itself wildly, going throughout your system looking for more of these virally infected cells or cancer cells, okay? Right, because this could also be cancer. Let's say you have a cancer mutation there, okay? Mutation that would cause cancer. Then what happens is you produce different proteins than you would otherwise produce. You serve up different protein fragments, okay? And really, there are only very few childhood cancers, okay? And those are operating in some different way. Most of them, we're getting all the cancers. Our little toddlers are getting all the cancers, and they're just being taken care of by this, okay? Okay, so what's the machine learning here? What I'd love to do is I would love to take that set of TCRs. You remember, I got that from adaptive. I said, oh, you have these 10 to the 6th T cell receptors that I've sequenced. I'd like to be able to take that set and say, oh, you have a certain disease or a certain set of diseases. There was a proof of concept with CMV, okay, which is cytomegalovirus, okay, where they showed that with 90% accuracy, better than the best diagnostics, they could just look at your T cell profile. And a lot of us are carrying this virus and say who is carrying it and who is not carrying it. However, this does not generalize well. As you might imagine, disease produces many different protein fragments, and so some of the one set of TCRs doesn't map very cleanly to a disease. Does for CMV, but usually not. So instead, we're going to look at the TCR and then this peptide that is being held here by the MHC. And we're going to ask, how well do they bind? Because that's the signal. When they bind well, that's the signal for the T cell to kill the cell and start multiplying. Okay, so how well do they bind? Well, usually protein-protein binding is a very difficult problem, just computationally it's difficult. In this case, it's not so bad because it turns out that the peptide sits right at this plane, and so from the structural biology point of view, that really cuts down the variation. And in fact, a couple of papers just came out, I believe in Nature and PLOS One or something, just in the last few months, where a couple of different groups, one here at Stanford, had 10 protein fragments, okay, and 5,000 TCRs. And from that, they were able to predict binding energy. So that was their training data. Okay, it's a matrix with 10 protein fragments, also called antigens. When these little proteins get presented, then they're called antigens. When they're cancer antigens, they call them neoantigens, but they're all the same thing. So 10 antigens and 5,000. Now, just remember, really, how many possible antigens are there? Even if they're only 9 long, it's 20 amino acids to the 9th. So that's like 1.8 times 10 to the 10. So there are 10 to the 10. They chose 10 out of 10 to the 10, okay? They chose 5,000 out of, in the human population, there are 10 to the 15th to 10 to the 16th different TCRs. You have about 10 to the 12th in you, okay? Which is why as a population, we do much, much better than as an individual. Okay, so we have, this is a sparse matrix completion problem. We have 10 to the 16th by 10 to the 10. They've taken 10 by 5,000, and they were able to say something, this was a big deal. But now, what do we have going for us in this collaboration? Okay, so we have, first of all, these are the things that can sequence the TCR. So every time you give me a little bit of blood, or a little bit of a sample from your tumor. So when you have cancer, we take samples from your tumor, if it's a solid tumor, and we look at it and we say, oh, there are tumor-infiltrating lymphocytes. We expect those to be very enhanced in whatever is going after this tumor. Okay, so you can look at blood, you can look at little samples from the tumor, and I can give you every shot 1 million TCRs. And then there's even something more exciting than that, an assay called MIRA. And what that does, and remember what these other guys did, was 10 antigens by 5,000. This does 1,000 antigens, 1,000 protein fragments by 1 million, and we can do this about 10 times a day. Okay? So now we have billions, on the order of 10 billion labelings a day. Now I do have to tell you, this is still a really hard machine learning problem because the data is very sparse. Of those million, only 50 to 100 will bind with any given antigen, and at very different strengths too. So this is a sparse problem. Okay, but that's the data we've got, and we're starting to develop models. Okay, so towards a universal diagnostic. T cells are nature's universal diagnostic machine, and this is not a new thought. TCRs, the T cell receptors, were discovered 30 years ago, and everybody knew that if we could understand what it meant that you had certain T cell receptors in you, we could really know what your body is trying to deal with to stay alive, okay? But the difference today is that we will be able to generate billions of samples a day. This is really cloud-scale machine learning. And what we hope at the end of this is to come up with empowered care, a universal diagnostic. And I just want to say one other thing. It's more than just, there's diagnostic, and then we're also going to be able to do therapeutics. And this is some of the work that we're doing with Stand Up to Cancer. What happens in cancers is that as they get more sophisticated, they release what are called checkpoints, which go and they sit on your T cells, and they don't let them function, okay? And our body has this possibility to help us with autoimmune diseases, but the cancers all seem to develop these things. The huge advances in cancer immunotherapy that, stage 4 melanoma used to be a death sentence, and now like 30% of the people are long-term survivors, is because we came up with checkpoint inhibitors, which kind of take this break off and let your T cells go back and kill the melanoma. It's also been fantastic on stage 4 lung cancer. It's first line now, first line therapeutics for stage 4 lung cancer. So, but it only works for like 30% of the people, a particular drug. So if we can understand what's going on in your immune system, I can say, no, don't try that drug on him. It's not going to work. Or it's going to have horrible autoimmune consequences. Try this drug on that person or that drug. So we'll be able to read your T cell profile and say, these are the drugs you should be on. And even more than that, in one of our projects, we've got a cohort of 60 long-term pancreatic cancer survivors, okay? Pancreatic cancer, kind of a death sentence, 4% survival after 5 years. We've got over 60 people who have survived over 10 years, and everyone believes that is due to their own immune systems having controlled the cancer. So we know the 6 TCRs that those people seem to have that other people don't seem to have. If we could figure out how to make a vaccine to get that into other people's systems, we could also have a vaccine to help to prevent pancreatic cancer. So, it's diagnostic, but in fact, it's more. I think this is going to be the way we deal with disease, a decade or two from now. Hopefully only a decade. Okay, thanks a lot.
Thank you, Jennifer. That was fantastic. We have time for a couple of questions.
You were a professor, I can tell you, before. I read your bio. I know you were a professor. Excellent presentation. Very clear, congratulations on that. I was on a board of a in silico small molecule drug discovery company. This ML, this kind of a thing, is that being used in the small molecule drug discovery?
It is not at the moment. I will tell you, I sit most of my time I'm in Plains, but I sit a lot of my time in Cambridge, Massachusetts, which is, the nexus for all the biotechs. And so many people are coming to me and asking me, can we do this same kind of thing? And what we're hoping to do, and I'm not much to, try to plug our company, but I think I really should right now. We are building very sophisticated tools into Azure so that we will be able to help people, to use these tools themselves as platforms for biotech.
Especially small molecule drug discovery, which will be ideal, and the cost will go down in the discovery.
Exactly.
Thank you. Good job.
Exactly. Yeah.
I wanted to follow on to that question in terms of, what else do you think we can do to have more of a, to borrow Andres' phrase, a kind of software 2.0 model where we can kind of democratize the collection of that data and the labels so that more people can have access and innovate on the gene therapy and the things you talked about?
Absolutely. So look, I really, that's why we chose the adaptive partnership. We said, where do we put our time so that we can develop platforms? We want to build from this with adaptive a healthcare platform that people can come in and use. Adaptive will still make money because they do the assays, okay? And you still got to send in people's blood. You still got to send in their tumor tissue. But this is going to be, so adaptive will do fine because they'll do your assays. We're gonna have all these tools in this platform built into Azure, and if you're an Azure customer, you're gonna be able to use it. So we'll have a healthcare platform. And so this partnership has a business model behind it that will democratize the use of this, because we all believe, the reason that I'm working on it, that we're all working on it, is because we want to get this out to people, because this is just this is what precision medicine means. It's about you. It's about your genome. It's about your immunome. And it's about the right care. So yeah, we really, that is our absolute goal, is to get it out there so that everybody can build on it.
Heather, thank you. Great talk, inspiring and really exciting potential. You talked about a couple of the challenges that face compute for ML and biomedicine, namely really large data sets and high degree of sparsity. What are the other challenges that you see for compute in this domain, and what would you do with orders of magnitude more?
With orders of magnitude more
compute horsepower.
Compute. Okay, well, I think there are, just like in the CRISPR case, we landed up pre-computing a lot, okay? So in this case, I mean, these problems are very hard problems. These are structural biology problems. We do transfer learning and things like that, but with greater compute, we can really nail it much, much better. So, there are going to be huge amounts of pre-compute here. And I also honestly believe that in other domains of healthcare, image stuff, there are places where DNNs are going to revolutionize it. There's, and this is other stuff that other of my researchers are involved in, but, natural language processing on electronic medical records, recognizing cancers and other things in images. And so those are also areas where, but that problem that I showed you, that structural biology problem where you have 10 to the 16th by 10 to the 10th, that many protein-protein interactions, we can use more compute. Okay.
All right, everyone. Thanks so much to Jennifer. Let's thank her once more.